So hello and welcome all. Thank you for joining us for our first ever live coding workshop for the Sciences and Parks program. My name is Jessica Johnston. I work with the Ecological Society of America, but I'm essentially an engagement partner with the National Park Service and the Conservation Legacy. So what that means is I get to host really cool events like today's, and I also support affinity groups and other types of engagement sort of events. Today, your leader of the workshop is a former professor of mine. She's an associate professor at Virginia Commonwealth University, Catherine Hulshof, and she's also a certified data carpentries instructor. So she's quite familiar with coding, and I will let Catherine introduce herself. Welcome, Catherine.
Great. Thank you so much, Jessica. I'm Dr. Catherine Hulshof. I'm an associate professor at Virginia Commonwealth University in Richmond, Virginia. I study forest responses to climate change in tropical and temperate mountains, and I teach data science for undergraduates and graduate students. I'm really excited to introduce you to some of the do's and don'ts and the basics of data science today. Do the other two helpers want to introduce yourselves?
Sure, I'll pop up first. Hey everyone, I'm Alexandra Julik. I'm a marine ecologist and a postdoctoral research scientist at the University of Florida. And I was a SIP fellow in 2021, also with Andrew, where my project, I was stationed at Buck Island Reef National Monument in the US Virgin Islands. And my project focused on manipulating and analyzing a really large data set on their sea turtle population in the park. So that kind of really got me into data science, honestly, was that project. So yeah, I'll turn it over to Andrew.
Hey, so I am Andrew Burchill. I was also, a SIP intern with Alex, and I'm now a data scientist with the city of Scottsdale. So I see some people are from Phoenix, so nice. But yeah, I also worked on a pretty big multi-year data set when I was in SIP, and now I kind of just try to get people in government to manage their data better, against their will. But yeah.
Great. So today we're going to cover two topics: spreadsheets for in environmental data science, and we'll also cover OpenRefine. So these are some of the basics. Now, in an hour's worth of time, you will not be the expert in data science, but what I want you to get out of this 1 hour workshop with us is that you're more comfortable using the tools to keep your data clean and tidy and be an incredible scientist. So with that, let me share my screen, and we're going to start with spreadsheets for ecologists. Before we begin, share screen, desktop. Okay, here we go. So we'll be covering material that is curriculum for the Carpentries. So the Carpentries are a globally recognized organization that designed curriculum in data science. What I love about their curriculum is that it's recognized by a lot of potential employers. So when they see that on your resume that you've completed some of these lessons, they usually know like what skill sets you were able to take away. And if you want to go into more depth than what we can go into today, you can refer to their curriculum and work through it on your own online as well. So those resources are available. Now let me adjust. I'm working off of two screens here. So let me get this going. I'll cover some theory and ideas before we get into the hands-on portion. So I want to start by talking about good data organization, because that is the foundation of any research. And most researchers have data or do data entry in spreadsheets. And the problem with spreadsheets is that we tend to work with data in a way that is very different from how computers handle data. And this can cause problems later when it's time for analyzing or graphing or sharing that data to collaborators or making it openly available. So in this lesson, we will cover how to think about data organization and good data entry practices. We'll also learn how to avoid common formatting mistakes, how to handle dates in spreadsheets, because dates are especially problematic in programs like Excel or Google Docs. We'll learn about something called quality control. I promise it's not as boring as it sounds. And then we'll learn how to export data from spreadsheets into a universal format. So let's start with an overview of good data entry practices. These are tips for making what we call data tidy. So there are 10 rules to follow when entering data into a spreadsheet. So you can imagine if part of your work during your internship will be entering field data into a spreadsheet. These are 10 tips that you want to keep in mind before you go into the field. So we want to be consistent. That's our first rule. This applies to how we name our columns, how we name the data entries. We could do this by making the data into a rectangle, and we'll see examples of all of these in a few minutes. What I mean when I say make the data into a rectangle, that means every row is a single data point, and each column contains only one type of information. So when we do this, we'll see how each cell in your spreadsheet, like in your Excel file, contains a single value. The sixth tip is to minimize redundancy and avoid the use of multiple tables. So in Excel, you have the option to have multiple tables or tabs in a single file. We want to avoid this because computers can't easily detect these additional tables. We also want to avoid the use of colors, fonts, or anything visual because computers don't see visual information the way that humans can. We'll also talk about null values and the difference between an empty cell and an NA. And we'll see how to save data in plain text files and why that's important. And finally, we'll learn how to use good names for column headings and for the files themselves and why this is important. So this is a broad overview of some of the rules that we'll be covering. And now we're going to switch over to Excel. So you should have opened or downloaded an Excel file. And I'm going to switch now to that Excel file called messy survey data spreadsheet messy. Now I'm going to zoom in to help you see the field. So before we get into the specifics, now would be a good time to get into our small groups, and based on those 10 tidy data rules, I want you to tell me everything that is wrong with these files. So remember, you can navigate to the file itself, explore the different tables, and I want you all to make a list of all the things that are wrong with these files. There we go. All right, back in the main room. Can you see the Excel file?
Yep.
Okay, thanks. All right, so I hope you got all of the 10 rules for keeping data tidy. Yeah, we had a great discussion of all the subtleties. So let's talk about formatting. So a common problem is creating multiple tables in one spreadsheet. Did that come up in the other breakout room too? That was one of the first things that we noticed, right? Having multiple tables in a single sheet confuses a computer. It should be avoided completely. And that's because a computer sees each row as an observation. And when it reads a spreadsheet, it assumes that all of the columns are related to the same sample. But you can see this row here is actually represents 3 distinct samples. Another problem that was brought up was these multiple tabs, right? We've got 3 tabs in a single file. And when you use extra tabs, the computer will not see the connections that are there the way we can tell that there's connections. And when you're going to analyze that data or collate all the data, it creates a scenario where you're likely to accidentally add inconsistencies. So for example, if you were copying and pasting, but you accidentally didn't copy this column correctly, it's just, it adds a point of vulnerability to your data. And another thing that was brought up was the blanks. Blanks are okay, but it's not clear if this was an individual that was caught and escaped, or why the data wasn't recorded. So a better practice would be to fill in those NAs. R reads blanks as NAs, and other federal institutions and state agencies, I think, use some strange number like negative 9,999 to indicate NAs, which is not a recommended practice for keeping data tidy, because you have to know what that value is. It's easy to forget about that value unless you're aware of it, and you may not be able to detect it until later on when you're analyzing the data and your statistics look weird, and it's because you have these negative 9,999 values, right? Those are real numeric values. Another common problem is using formatting or anything visual to convey information. Highlighting cells is something that I see a lot of early career students and trainees do, but this should be avoided because a computer can't detect visual information the way that humans can. So instead, if you need to flag this cell for some sort of information, create another column, and that might be like something like notes, or here something about calibration, and then fill in not calibrated correctly for those cells. We also don't want any metadata in the file. What I mean by metadata is descriptors like field season or plot. Like, this should be its own column, and there shouldn't be any space between the column names and the data itself. And that's because when you go to read in this file into a program like R, it expects the first column to be the header or data. And so when you have these extra columns, it really doesn't understand where the data begins. Finally, let's talk about names. So we want descriptive names, and this is because your future self or your collaborators will not understand what you mean by variable 1 or variable 2. So instead, we want to pick a naming structure that's descriptive, but still concise. And it's important, unfortunately, to avoid special characters or spaces, because programming languages like R are confused by these. And this is really unfortunate because a lot of non-English languages use special characters. And so that just shows you the kind of the language bias inherent in these programming languages. All right, the next rule is dates. Dates are very special in Excel. So it's tempting to store dates in a single column, but that is not the best practice because dates are stored in a problematic way, even though in programs like Excel or Google Sheets, they're displayed correctly. So in Excel stores dates as numbers, as the number of days from a default date of December 31st, 1899. Like, so random, right? Like, how would you know this unless you came across some obscure literature about that particular default date? Another issue with dates in Excel is that Excel turns things that aren't dates into dates. So G names like MAR1, MAR1 is converted to March 1st. And there's a really great famous scientific article about how many problems there are in these genetic studies because of how Excel handles dates. So instead, it is much safer to store dates in multiple columns for year, month, and day. An alternative is to store your date in a universal format, something like, it might be date, and then just like you would give the unit, the 4-digit year, the 2-digit month, and the 2-digit day. If you really need to store the date in a single column, so that would look like this. And that way, when you go and read this file into a program like R, it'll read this column as an integer, and then you can specify, well, this column is a date, so read it with this format. Another reason why specifying year, month, day in separate columns or specifying the format is because this varies. The way we display dates varies across countries. So some countries will write, let me give you an example of this. Okay, let me see a good example. This is a good example of this cell here. So in some countries, this would be read as December 11th. And in other countries, this would be read as November 12th, right? Depending on the country, whether the day is displayed first or the month is displayed first. And we want our data to be universally understood. And so we want to specify your month date in separate columns or specify the format that you're providing your date in. Okay, now we're on to quality control. And yeah, we're on time. So I'll give you a quick example of quality control. These are great when you're entering data in the field. So there's two methods for ensuring that your data are high quality and error-free. The first is called quality assurance, and the second is quality control, or this is QA/QC. So quality assurance happens before data is collected in the field or in the lab and prevents bad data from being entered in the first place. And then quality control happens after the data is collected and it checks for bad data. So in this segment, we'll look at how to set something called data validation rules in Excel that will check for obvious errors. So let me set a data validation rule for this column. And I'm going to pretend that we've tidied this up. So to tidy this up, I would have to get rid of this. And I would have to do this real quick so that each cell is a single column, not calc, not calc. Let's get rid of this. Let me do the species DO, and I'll only do it for this second table so we get a good idea. Okay, now I can delete this. Delete. Maybe I want this as year, and then month. Let's see if I can do that. Year, month, day. So I'd have to go year, month, 7, 16. Okay, to give you an idea of the process. Oh, we need to get rid of this. Replace the unit with nothing. And then let's specify those units in the column headers. And then these need to be deleted. Okay, so you can get an idea of how we would tidy up this data a bit, how we would convert the date to year, month, day. And now we can set our validation rules. So usually you select a single column and it's data. I'm using a program on Linux. It's a little different, but it still should be under data in Excel. And there should be something called data validation or validation. And we want to select validation so that we get this pop-up menu. And we want to allow...
Just a quick thing. We can't see the pop-up menu.
Oh, darn. Okay. Maybe...
Because it probably opens in a new window, so and only this current window is being shared.
Desktop. How about now?
Perfect.
Okay. Okay, great. All right. So for the different, thanks for that. For the different validation criteria. So let's say, you know, I've been doing these surveys. These are small mammal surveys collected from Arizona for the past 40 years. And based on this 40-year data, I know that our measurement tool only reads in whole numbers. And I want data between, I know that we, based on 40 years of data, all the individuals we've caught are greater than 1 gram, and they're below maybe 500 grams for like these big kind of desert rats. Okay, and so now when maybe I'm using a tablet to enter this data in the field, and I'm entering something, and maybe my pinky slips or my tablet slips, and I go to enter 5,000 when it should be under 500, I get this error message to double check. You can, in Excel, you can also customize the error message, like double check the value should be between 1 and 500 grams. And so then it's a flag to the user inputting the data that, oh yeah, my finger slipped, it should be 500. So that's one example of quality assurance. Quality control happens after data collection, and this usually happens in R. So if you go to next week's, we can see an example of that. But really, you're looking for the min, you're looking at the summary of data. So you might look at the min and maybe the max to see if there are any, oopsie, equals max, to see if there's any wonky values. So that's one more basic way for quality control, just to check for values that should be reasonable. All right, so now we're ready to export our data. We've done all our quality assurance, our quality control, and we always want to save our data in non-proprietary formats. This is because of a few reasons. One, Excel, you have to pay for Excel, right? Not everybody has access to paid software. Two, even though Excel is fairly common now, in 100 years, 150 years, when someone is going back to your data and doing historical analyses, Excel might not exist. And so we want to preserve the data for longevity. And the easiest way to do that is to save it in a format that is much more universal, like a .txt or .csv. So in ecology and environmental science, you're more likely to work with .csv or comma separated values, and you can export menu, save as. Do you see the pop-up window where you can select the comma CSV format and save? All right, so now we have our data from the field, and I want to switch to OpenRefine. Now I'm in that, let's see, present, okay. So now, right, maybe it's the middle of, you've just come back from the field, you've done all this data input, and now you want to check for potential errors. So this could be another example of quality control that happens after a data collection. So most ecological data is generated by humans, and we make mistakes, right? Which is why a major part of the data workflow is preparing the data for analyses. And a lot of this involves data cleaning, where errors in the data are identified and corrected, or formatting made consistent. So in this tutorial, we'll work through, again, the carpentries tutorial on OpenRefine, so that you have that resource you can consult with throughout the summer if you need to. OpenRefine, it's free, it's open source, specifically for working with messy data. And there's nothing wrong with messy data. Like, it's very normal to have messy data, and cleaning the data is part of a standard kind of workflow procedure. What I love about OpenRefine is that it saves editing by hand, and we want to avoid editing anything by hand. We want to automate as much as we can, because that saves time, it reduces errors, and it makes you a better scientist. A lot of what we're going to see today can be done in R, but if you're new to R, OpenRefine is a great tool that'll get you there faster than trying to figure out what these code errors are. So we'll see an example of cleaning data in OpenRefine. So why use OpenRefine? Why clean data? Well, some journals and granting agencies require documentation of the steps that you took when going from raw data to clean data. And OpenRefine captures all the actions applied to your raw data. With OpenRefine, you have to export a modified data set to a new file. And this is part of that tidy data tips, is keeping the raw data separate from your modified data. And the data is often very messy, and this tool saves a lot of time, especially if you're working with very large data sets with thousands of rows where you physically cannot edit each cell by hand. Data cleaning is also often very repetitive, and so you often find yourself wanting to perform the same cleaning steps on multiple files or multiple cells. And OpenRefine is perfect for speeding up those repetitive tasks. And in our help sheet, there's a link for more information on OpenRefine. There's an entire OpenRefine community, a wiki, a Google group where if you want to do something very technical and granular, there's probably somebody who knows how to do it. So let's start by creating a project in OpenRefine. So take a minute to navigate to where you stored the OpenRefine folder. I store it in my documents because it's a place where I don't keep many things, and so it's easy to find. And I'm running Linux, so mine is going to look a little bit different. Or look out for that diamond icon or that .exe, and I'm going to run this file as a program. And hopefully, right, like this terminal window opens, and hopefully you'll be able to see this new tab opening in my browser.
Yes, we can see it.
Okay, thanks. All right, so for this second part of the tutorial, we're going to navigate to that second file. It was called this portal_rodents. All these digits. Go ahead and open. And there's a lot of steps here. So having that help sheet open is helpful if you miss a step. It outlines kind of all the menu options. Next, so we're right here. Choose files. We're going to select next and create project. So next. And it should upload and everything looks okay. If it doesn't, if it looks strange, you might have to select the appropriate separator. This is a comma separated file. And so, yeah, this looks like what a spreadsheet should look like. And I'm ready to create the project.
In case anybody's having a hard time following along, you're welcome to ask questions in the chat. Andrew and Alex will try and assist you.
Yeah, thank you. Because there are some steps here, and I recognize it can be challenging to follow the Zoom video while you're switching back to your browser window as well. Okay, so let's talk about faceting, because facets are one of the most useful features of OpenRefine. And it helps give you an overview of the data and bring consistency to your data. What a facet does is it groups all similar values in a column and then allows you to filter the data based on those values. It allows you to edit values across many records at the same time. So one type of facet, and probably the type that people use OpenRefine most often for, is a text facet. So let's see an example of a text facet. If you find the column called scientific name and select this down arrow in the column header, you will see an option at the very top called facet, which will open another menu, and the top option, at least on my version, is text facet. So we're going to group the scientific names using this text facet, and what you see in this left-hand window now is all the groups. You can already see maybe some spelling errors, right? Maybe these three should be lumped together. Yeah. And you can sort these facets by name in alphabetical order, by count. So this is the number of cells that have this particular entry. I like, going back to the name, you can see these top two. They look almost identical, but something about the entry, a computer is distinguishing these two entries as different. So let's see.
The difference is a space. I realize this just reminds me of my first paper I ever published. I did the analysis technically incorrectly. I only caught it like several years later when I was looking at my data, and it was I had broken it. I did this and it was horrible. I'm sorry.
A real life example of how spaces are impossible for humans to detect, but much easier for computers to detect. So we're going to use OpenRefine to detect it for us, so we don't have to. Thanks for sharing. All right, so we'll start to see some problems. And so we've seen some problems already. Another feature of OpenRefine is this cluster option. So, you know, there could be hundreds of species. We don't want to go through each one. We want OpenRefine to do the work for us. So if we select the cluster button here, there's different methods of clustering. To be honest, I'm not sure like what they're, you know, they might detect different capitalizations or different ways of clustering text. So I'm going to do key collision and the keying function. I'm going to select metaphone 3. So I can see like these are all of the entries that OpenRefine is saying should be clustered together. And then I can decide, yeah, these are all spelling mistakes. You know, maybe my field technician entered an E instead of the A. I should have had a quality assurance in my, do a data validation for that column. And yeah, these all look good. So I'm gonna merge selected and close. Oh, you must select merge. You must select which ones you want to merge selected and close. And so OpenRefine edited 739 individual cells that had spelling mistakes and errors, right? All right, so sometimes data, another feature we'll see is splitting the data. Sometimes data in a single column needs to be split into multiple columns. And if there's a common separator, like an underscore or a space, that helps, we can use that separator to divide up the pieces of the column. So let's say we want to split scientific name into separate columns for genus and species. And we can navigate to this down arrow at the column header for scientific name. And there should be something called edit column. And the first option in that submenu is split into several columns. So I'm going to split this. And I want to choose the separator by a space. So I'm going to enter a space. You can't see it, right? But it's there. And I'm going to select OK. And it does its thing. Oh, this is interesting. Because now there are 1, 2, 3, 4. Where I expected 2, there are 4. And that's because somewhere before the letter A in this example, there were 2 spaces that we couldn't see. So OpenRefine is splitting based on those spaces. So let's see another feature of OpenRefine. I want to undo what I just did. I want to undo this split. If you select this undo redo tab in the upper left of the window, you can see all of the edits that you've made. And if you select on, 0 was create project, 1 was we edited the scientific name column. We did that cluster. If you select the 1, then the data goes back to the state it was in before you did this split, right? So I want to select this and apply. Oopsie. No, that's not it. Okay, you don't have to select anything, right? You clicked where you want to redo to, and it gives you the state the data was in prior to that action. So now I want to clean up these spaces. Another useful feature of OpenRefine is its ability to trim leading and trailing white spaces. So these are spaces before and after names or values in each cell. This is really important because, as we heard in the example, words with spaces at the beginning or end are hard for humans to detect, but those blank characters make a difference to a computer. And in most cases, we want to remove these. OpenRefine makes it easy to remove blank characters from the beginning and end of entries. So in the header for the column scientific name, again, we're using that down arrow. We want to now edit cells, and we want to select common transforms and trim leading and trailing white spaces. And in just one click, we fixed, I guess there were 3 cells with those hidden white spaces that were hard for us to see. So now we're ready to split that scientific name into genus and species without those white spaces getting in the way. So we can see an example of that. Where was it? Where was my split? Edit cells, edit column, split into several columns. And I want to select my separator as a space. And I'm going to uncheck remove this column so I can see. Sometimes I like to keep it. Great. And now we've done that split, and there's only 2 columns, so that tells me that there were no hidden white spaces. All right, how are we on time? We have about 10 minutes. And I will, let's see a few more features. So another useful feature when you're working with really large data sets, sometimes you want to work with just a subset of the data. To do this, click the down arrow next to scientific name. And we want to do a text filter. So we can filter based on any character sequence, maybe something like BAI. And so what OpenRefine is searching for any character sequence that matches BAI in any part of the entry. So it gives me 48 matching rows that have BAI in the entry. And we can scroll down. We can look at all 50. I'm going to select show 50 rows. And if we scroll down all the way, we can see that 2 species match that filter. And we can also see that here as well. That was nice. And you can see how many, what the count are. And maybe that's because, I couldn't remember what the species name is, but I know it starts with BAI. So let me look that up. So faceting gives you an overview of all the data, while filtering allows you to select a subset of that data. Just like you can in Excel, you can also sort your data. Sorting in Excel is a little risky because if you don't select all the columns, there's a huge risk that you could be separating rows. And that is a problem that is impossible to fix once you've done it and saved the file. It's hard to detect that. But doing it in OpenRefine is less risky because it knows to maintain the integrity of that row. So it will never mix rows. So let's talk about sorting a column. Let's go back. Let's remove this filter so we can look at all of the data. We can sort on a specific column based on text. And maybe I want blanks at the top. I don't think there's any blanks, but you can move these around. Maybe you're working with, maybe you want to see all the valid values are at top or the NAs at top, and we're sorting cell values as text, but if you had numbers, you can do that as well. And so it sorts the column by in alphabetical order, and it maintains the row integrity. And in the next 5 minutes, there's one feature of OpenRefine that is really cool, will really impress your boss. So I think it's worth mentioning. It's the ability to export scripts. So OpenRefine, like we saw in the undo redo, OpenRefine is saving every change that you make to a data set in a format called JSON, JavaScript object notation. Like I don't know JSON, but I do know that I can export the script that OpenRefine uses, and I can open up a new file, a separate file, and run the same steps as long as they have the same column headers. So if you had 20 files to clean, and they all potentially have the same types of spelling errors and white spaces on the scientific name column, then you can save this JSON script, open a new file to clean it in OpenRefine, paste in that script, and run it. And so in this undo redo, that's where we export our scripts. So if we want to extract our script, this is the script that it uses, and it's already copied, so you could copy, open it in a text file, and paste it. And then when you go to open up a new file, maybe you had, portal rodents 2003 to 2023, then you would copy and paste it back into here, and it would do the same steps, which I think is really cool if you're working on multiple files. So that's the last feature. The very last feature is, okay, now we've cleaned our file, let's export it, right? So we want to export, remember, we want to save it into a non-proprietary format. That's typically going to be CSV file, or these comma separated value files. And it'll append this -CSV file, which is, it makes it a little easier to rename. So you might want to rename this something like cleaned, maybe with the date 05/17. That way we're keeping our raw data separate from the clean data. And that's it. Those were the overview. I can stop sharing. An overview of keeping your data tidy. We saw some tidy data practices in Excel, in spreadsheets, and then we also saw some quality control in OpenRefine. So I hope you found this useful. I hope you learned something that you can use in your internship and really impress your boss of what a great data scientist you are. I'm happy to take questions.
Yeah, we have a couple minutes for questions. You can raise your digital hand if you have a specific question that is unanswered. I know that was super fast, but that's the reason why we're recording it, so you could go back and look at it over and over again if you need to. Actually, Catherine, I have a question for you. So I saw that you could split columns, but can you join columns as well? Like if you, for example, I have a data from a survey that it outported it or it exported it as like numeric across, and I wanted it collapsed in a single column. So OpenRefine can do that, I'm assuming.
Yes. Yeah, it can do, OpenRefine is a little, is a lot like Excel. I mean, some of the things that if you're familiar with doing in Excel, you can do in OpenRefine. And what's great about OpenRefine is that it saves the steps and it maintains your data integrity. So there's no risk like there is in Excel of overriding a cell, or, you know, that there's an error message in Excel that pops up when you're splitting cells, like data is still in cell. Do you want to overwrite? And you might accidentally press yes and not mean it. So you can edit cells and join multi-valued cells. And you'd select the separator. Like maybe you want, let me see if there's a way. I've never done it. I know there's that edit cells, join, but it would be edit. Here we go. Join columns. You see where I found that? Scientific, okay, column, edit column, join columns. So I want scientific name 1, what was it? And scientific name 2, where are you? And the separator, maybe I want an underscore. And now it creates that new column.
Awesome. I see Kyle's got his hand raised.
Yeah, question. If one of your data like entry points is initials of people and one of someone's initials NA, would you then go with a blank or another filler?
You're muted, Catherine.
Okay, I see 2 solutions. It's use a 3-letter initial, or when you're going to enter that data into R, then specify that that column is a text column and that an NA value is something else. Like maybe it's NA. And there's a way to do that in R so that yeah, it doesn't identify that as an NA. Great question.
Well, I want to be respectful of everybody's time here. I know we're at 1 hour. So I don't know if Alex, Andrew, or Catherine has any time beyond the hour. But if there's any remaining questions that got unanswered today, you can reach out and email us. I'll drop the SIP engagement email in the chat. And I want to also say special thanks to Catherine, Alex, and Andrew, because they do all have their own jobs and none of it is related to scientists in parks now. So thank you very much for taking time out of your day to do these trainings. I hope you got a lot out of this. We have a survey that's gonna prompt as soon as you hang up from this call. So I would appreciate if you could give us feedback. This is the first time we've ever done anything like this. And hopefully some of you will see next week for part 2 of the coding, which will be actually doing some work in R on data visualization and really starting to jump into the coding language that is seems to be the future, right? So thank you all, and feel free to reach out if you have any questions. And
Thanks,
happy
everyone,
data
and good
wrangling.
luck this summer. I'm excited for you as you start your data journey.